Last but not least, Dr. Alfredo Martinez is the director of the Biotechnology unit, part of the R&D department at Mesoestetic Pharma Group. He has a PhD in biomedicine and possesses a very broad experience in the study of hyperpigmentation, anti-aging, and epigenetics. So, for our final presentation, over to you, Alfredo.
Okay, thank you very much for the introduction. I am Alfredo Martinez, and I work for Mesoestetic Pharma Group in Barcelona. So one of the main fields of research in our company is skin aging. We are constantly doing research in order to test active ingredients that we later on include in our final products. In this sense, lipid droplets seem to be altered in the damage that happens after advanced glycation in products, which are one of the main drivers of skin aging. So this is very useful for us because we can use it is very useful for us to have a software that can quantify lipid droplets dynamics in order to test the efficacy of these active ingredients that we are going to use in our products. So in our experiment, after inducing the damage with glycation end products, we observed an increase in lipid droplets area and density. These findings correlate with evidence from other studies that report that lipid droplets accumulation occurs in multiple cell types after this type of damage, the damage that happens with advanced glycation end products. The high temporal resolution in this technique suggests that this lipid droplet accumulation occurs after it does not start immediately, but there is a lag time after which these morphological changes occur, which is 6 hours. After this point, the accumulation follows a linear trajectory. So as proteins are the main target of advanced glycation end products, we think that it is possible that these glycation end products target first these proteins and then bound to lipid droplets. So this could explain this lipid droplets accumulation and after the 6 hours, after this lag time. So the next step was to test if a protective cocktail of active ingredients could reverse these changes in lipid droplets. We did this, but unfortunately the cells went out of the field of view, so we were not able to quantify these changes. So we are going to repeat the experiment, or maybe consider performing this experiment using the automated microscope in order to get a greater field of view. So this is the first method that we use in order to quantify or to study lipid droplets. But we think that the great advantage is to the ability or the capacity to analyze live cells without using any specific fluorescence marker. So this is going to be very useful for us, as I was saying before, in order to study the efficacy of the active ingredients in the lipid droplets dynamics. And from a broader point of view, I think this is going to be very useful in order to study how these lipid droplets changes occur in different or specific pathologies or conditions, not only in the skin, but in other organs from the human body.